Hello my dear students and welcome to Zenith Academy Online. My name is Shagufta Jaipuri and today I'll be teaching you part 3 of the circulatory system, that is the white blood cells. This is for ICSE class 10 curriculum. Students, we already know the composition of blood from our part 1 and we know that white blood cells and platelets together constitute about less than 1% of the blood. In our last lecture, we had studied in detail about red blood cells and today we are going to study about white blood cells. So as I already told you, the white blood cells account for only about 1% of our blood, but their impact is very big. The white blood cells are also known as leukocytes. See, there is a spelling Leukocytes and also Leukocytes. So both of them means the same. The leuko means white. Now children, the WBCs have a nucleus. In our last lecture, we studied that red blood cells are without a nucleus. They are enucleated, but WBC have a nucleus. So this is one of the major differences between both of them. Now their number. The number of WBC is much less, which is usually about 4,000 to 8,000 per mm³ of blood. In RBCs, we had studied the number of RBCs are around 4.5 to 5 million per mm³, but here the count of WBC is only 4,000 to 8,000 per mm³. Here you can see a process known as Diapedesis. Here you can see that this is a blood capillary and this WBC are able to ooze out of it. So wherever they are required, they just can move out of the blood capillary and can go to the tissues. This process is known as diapedesis. I have given you one more diagram. So this is the blood capillaries and these are the WBCs which are able to squeeze out of the blood capillaries and go to the tissues wherever it is required. So most WBC are amoeboid, that is they can change their shape and can produce pseudopodia. Pseudopodia means false legs. Pseudo means false, pedia means legs. So they can form their false legs. You can see this is squeezing out, this is forming a leg. And they can squeeze through the walls of the capillaries into the tissues. Diapedesis, where dia means across, they can move across the capillary, and pedesis means oozing out. So students, there is a major question on diapedesis asked every year in the board exam. So you should go on Google Images and find out all the images of diapedesis and understand it very well, because the question on diapedesis mostly comes from a diagrammatic question. Okay, so you should know, you should be very familiar with all the diagrams on diapedesis. Now children, the WBCs also protect you against illness and diseases. So they flow through your bloodstream to fight against viruses, bacteria and other foreign invaders. So when your body is in distress and a particular area is under attack of germs, then white blood cells rush in to help destroy the harmful substances and they prevent the illness. Now children, WBC is of two types, granular and non-granular. In granular, there are neutrophils, eosinophils and basophils, whereas the non-granular or agranular are classified as lymphocytes and monocytes. So first we'll study in detail about granular, that is these three. So here you can see that granular WBC are also known as granulocytes. So there are three granulocytes: neutrophil, eosinophil and basophil. Cytoplasm of these contains granules. You can see there are granules in the cytoplasm. So cytoplasm contains granules of enzymes which help to digest the invading microbes. So when there are microbes coming in, these granules of enzymes, they help to digest it. Now granulocytes account for about 60% of the WBC. We know in blood around 1% of WBC are there, and in that 1% the 60% of it is granulocyte, that is neutrophil, eosinophil and basophil. Now nucleus usually restricted into lobes. If you see the nucleus here, there are different lobes. You can see 3 lobes here, 2 lobes here, and this is indistinctly lobes. Now let us understand each one of them. First we go on to neutrophil. Neutrophil accounts for around 62% of the WBC. Now nucleus is with 3 to 4 lobes. If you see here, the nucleus is 3 or 4 lobed, right? And the cytoplasm is granular as usual. The neutrophils always stain with neutral dyes. So when you want to see the neutrophil under a microscope, you will have to first stain it with neutral dyes, then only you will be able to see them. So what is their function? The function is to engulf bacteria by a process known as phagocytosis. I'll explain you phagocytosis further when we go in the video. Now where are they produced? They are produced in bone marrow. So neutrophils constitute about 62% of the WBC. The nucleus is 3 to 4 lobed. They have a granular cytoplasm and they are stained with the help of neutral dyes. Their function is to engulf bacteria by a process known as phagocytosis, and they are produced in bone marrow. Now let us go on to eosinophils. They constitute about 2.3% of the WBC only, but their numbers increases drastically whenever there is some allergy in your body. Now here you can see the nucleus is bilobed. You can see it has around 2 lobes, and as usual the cytoplasm is granular. Also you can see that it is stained red. So it gets stained dark red with eosin dye. So if you want to see it under a microscope, you have to put which dye? Eosin dye. Now this eosin is an acidic dye. Earlier we studied that neutrophils are dyed with the help of neutral dyes, but here eosinophils dyes with acid dye, which is known as eosin, and that is why we are able to see them, and that is why the name eosinophils. Now their function is to engulf bacteria and they secrete antitoxins associated with allergy. So going ahead, I'll teach you how antitoxins work. Now they are produced in bone marrow again. Coming up on to basophils, they constitute around only 0.4% of the WBC. Here you can see the nucleus is large, okay, and we are not able to distinct it into how many lobes are there. So it is indistinctly lobed. The cytoplasm is again granular, and now here it is stained with basic dyes like methylene blue. Their function is to release chemicals, that is especially histamine, for inflammation which dilates the blood vessel. Going further, I am going to explain you what inflammation is all about. Now they are produced in bone marrow. So children, in granular, we studied about neutrophils, eosinophils and basophils in detail. Now we will go to non-granular, that is lymphocytes and monocytes. Now non-granular WBCs are known as agranulocytes. You can see here that cytoplasm doesn't contain granules. Okay. So cytoplasm is without granules and a single large nucleus is there. You can see here they are not lobed. So there is a single large nucleus out here. Now let us study lymphocytes in detail. Lymphocytes constitute about 30% of the WBC. Here they are the smallest of WBCs and you can see a single large nucleus out here, right? And there are no granules in the cytoplasm. Their function is to produce antibodies. They are produced in bone marrow and lymph glands, that is spleen, tonsils, etc. Let us study about monocytes now. They contribute to about 5.3% of the total WBC count. Now if you see here very clearly, this is kidney shaped, right? So nucleus is large and kidney shaped. Now at the site of infection, it transforms into macrophages. Macrophages are the one which phages means to engulf, to kill, to destroy. So they transform into macrophages at the site of infection. When I'll be teaching you the functions of WBC, I'll take this in detail. Now, their function is to ingest germs, and they are also produced in bone marrow. So we have studied all the types of WBC. Now let's go on to the functions of WBC. So now the first function is phagocytosis. This is the process in which most of the WBC and particularly the neutrophils, okay, they engulf the particle-like solid substances, especially the bacteria. This is a defense mechanism against disease germs. So an abnormal increase in WBC count up to about 50,000 or more per millimeter cube indicates that there is some infection, and that is why they have increased in number, and especially the neutrophils will go and engulf these bacteria. You can see here, I have explained phagocytosis here in detail. So here, I already told you that WBC are amoeboid in shape. They can form pseudopodia. So whenever a bacteria comes, then this forms false legs, that is pseudopods, and engulfs the bacteria inside. And then the enzymes in the WBC act upon them and they convert it into simpler substances. They absorb things and then they release their waste. So this is a process known as phagocytosis, and neutrophils are the ones which perform this function the most. Now going on to the second function, that is inflammation. Now inflammation occurs due to the reaction of the body tissues to the injury. So whenever there is injury or invasion of germs, so inflammation occurs due to the reaction of tissues to injury and to localized invasion of germs. Localized means at a particular place the germs invade, maybe due to a cut or something. The inflamed spot has several characteristics. Now how do you know that spot has inflammation? It has increased local heat, means it becomes slightly heated, that area becomes slightly warm, redness, swelling and pain. So these are the characteristics of inflammation. Now here the leukocytes, especially the monocytes and neutrophil. Which ones? Monocytes and neutrophils. Now they migrate through the walls of the body vessels. By which process? We already studied, by diapedesis, and fight against disease causing germs. They also destroy the damaged cells by phagocytosis. I already told you what phagocytosis is. Now, pus is mainly composed of dead white blood cells together with the tissue cells destroyed by the bacteria. So, when bacteria had entered in that tissue, it killed some tissues. So, there were some dead tissue cells and also the white blood cells which died in the process. So, pus is mainly composed of these two things. Now, let me explain you inflammation with a diagram. Now, here you can see that from the capillary, this WBC through the process of diapedesis came out to the tissues. Okay. Now here you can see that there is an injury and germs have entered. So the leukocytes, that is the WBC, act upon them, they fight with them, and then the dead bacteria and the tissue, dead tissue forms pus. This process is known as inflammation. Now coming to the third function of WBC, that is formation of antibodies. Now WBC, especially the lymphocytes, okay, they produce antibodies. Antibodies are those bodies which are produced to kill toxins or germs which enter the body. So they produce antibodies which kill or neutralize the germs or the poisons from them. Introducing the weakened germs or germ substances, that is vaccines, during the vaccination stimulates the formation of a particular antibodies which at a later period would destroy the particular disease causing germs if they enter into the body. Now students, you should know that what is vaccination. So whenever a vaccination is given to us against any particular disease, what is that vaccination? That vaccination is weakened germs or germ substances. So for example, if you are given a vaccination of chickenpox, then in your body the weakened germs of chickenpox are introduced. Why the germs are introduced? So that in your body responds to it and produces antibodies for fighting with these chickenpox bacteria that have been introduced. So these antibodies remain in your blood for a longer period of time, and when later in your life, whenever that chickenpox germs enter your body is ready with the antibodies to fight with it. So that is why vaccination is given. So vaccination is basically introducing the weakened germs of a particular disease in your body, so that your body is prepared and ready to fight for it whenever that germs attack. So now let us revise again antibodies and antitoxins. So when disease causing germs gain entry into our bloodstream, they produce poisons, and they produce poisonous substances, and these poisonous substances are known as toxins. Now in response, the WBC of our blood produce chemical substances called antibodies. So they are anti of the germs that have entered, anti of the bodies that have entered. So they produce chemical substances called antibodies which circulate freely in our blood plasma. Now the antibodies are specific to the toxins for which they are produced. So it is like a lock and key mechanism. The key will enter and open a particular lock only. The same way, a particular antibody acts on a particular toxin only. So the antibodies are specific to the toxins for which they are produced. So antibodies act as antitoxins which neutralize or detoxify the poisonous effects of the toxins. The antibodies may persist long after the disease has been overcome, and the person who has recovered becomes immune to the disease. So that is why they say that once you get chickenpox in your life, now you will not get chickenpox, because the antibodies that had been produced at that time persists in your body for a longer period of time, and then later you will not be attacked by that. So antibodies may persist long after the disease has been overcome, and the person who has recovered becomes immune to the disease. Now the disease will not occur to him. This is the underlying principle of vaccination. That is a deliberate introduction of weakened disease germ. Means you put the disease germ deliberately into your body. Why? To stimulate the production of specific antibodies. I hope you have understood antibodies and antitoxins very well now. Now let us study about the origin and life of WBC. Now white blood cells are mostly made in the marrow of the bone. So here you can see a long bone, and in this marrow this WBC are produced. Now they are stored in our blood and lymph tissues. So once they are produced in the bone marrow, where are they stored? They are stored in blood and lymph tissues. Now the WBC is also produced in red bone marrow, lymph nodes, and sometimes in liver and spleen too. The average lifespan of a WBC is 2 weeks. We studied in RBC that their average life was 120 days, but here the average life is 2 weeks. Now also, because some white blood cells have a very short life of around 1 to 3 days only, so our bone marrow is always in a process of making WBC. The neutrophils live for only a few hours, okay? And about 125 billion neutrophils are produced each single day. The old and worn out WBCs are destroyed in the same manner as RBC. Do you remember in RBC they were destroyed in liver, spleen and bone marrow. Now let us study the diseases. First is leukemia. It is a cancer of tissues forming WBCs whose number increases many folds at the cost of RBC, and it is usually a fatal disease. Now here you can see in the diagram that in the bone marrow, okay, normally the stem cells form red blood cells, they form granulocytes, A granulocytes, WBC, they form platelets and all those things. But in case of this disease, what happens? Only WBCs are produced and red blood cells are not produced. So they are formed at the cost of red blood cells, and this is usually a fatal disease. The current treatment of leukemia is only blood transfusion, nothing else can be done. The second disease is leukopenia. It is an abnormal decrease in the number of WBC. So that was all about WBC. I hope you understood everything about WBC in detail. So please watch our next videos for blood platelets and clotting of blood. Also the consecutive videos for detailed explanation of the circulatory system through heart. If you have liked our video, please give it a thumbs up and write in the comment box. And if you have still not subscribed to Zenith Academy Online, do it right away and don't forget to tap on the bell icon. Also share our channel with your friends and keep the learning on. Thank you.